All right, so what we have here is our solidified gel, and so the comb has been pulled directly out, so you want to make sure that that comes straight up. And then I'm going to pull my plastic thing here that has a bunch of that gel in it solidified. And so sometimes this can be a little tricky because it sticks. And so we're going to pull it straight up as well. And then sometimes that gel gets stuck on the bottom, so I just kind of wipe that away. And then I'm going to take this and I'm going to match up the wells to my machine. And so I'm just going to put it directly in there. Okay, so I have the gel now positioned inside and the wells are matched up. And so what I'm going to do is I'm going to take some of this buffer. And what buffer is it's kind of like an electrolyte solution that is going to allow electricity to pass through it. So the electrophoresis machine, as I mentioned before, has a negative and a positive side. And it's going to move from the negative to the positive. And electricity is going to be passing through these DNA pieces that I place inside. So this is the buffer. And so I'm just going to pour it into one side. And then once it starts to go over, I'm going to pour it in the other side. And it's got to go over the top. So you want about 135 milliliters of this liquid, like so. All right, so we're going to take you through the scenario now. And so what we're looking at, the scenario that comes with this kit talks about a young lady named Jillian. And Jillian is in a biology course. And what she finds out is that she does the PTC testing, and she finds out that she is not a taster. But every single person in her family is a taster. So because of that, she is sort of curious to figure out why is it that she is not, but they are. And so what we need to sort of figure out is a couple of members of her family to be able to tell whether or not they are tasters, heterozygous dominant or homozygous dominant. So we're trying to figure out exactly what they are based on that. So what I'm going to do is I'm going to take DNA samples and I'm going to add them to my gel. And so the first one that I'm going to add is the mother. So I'm going to go ahead and show you a chart here real quick. And so you can see that the mother is going to be in my first well, which is the well on the far left, and then dad, Jillian, her brother, her other brother, and her grandfather. And so we're going to put those into the gels one at a time. So I'm going to be using a micropipette set to 10. And so I'm going to go ahead and pull a sample out of mom. And we're going to go ahead and push that in, pull my sample. And so this is 10 micros, which is a very small amount. Okay, in the first well, I'm going to be putting mom. Pop the tip. Then I'm going to go to the second one. This is going to be dad. Same amount, 10 micros. Next one is Jillian. And then brother number one. Brother number two. And grandpa. All right. We are going to go ahead and turn on the machine and run it for approximately 25 to 30 minutes, and we're going to see what we come up with.